In this video, we are going to continue the process of glycolysis. Few things that you need to know before we study the further steps of glycolysis are: firstly, whenever there is oxidation or reduction of NAD positive and NADH, which means when hydrogen is added to NAD positive and it is reduced into NADH, or when hydrogen is removed from NADH and it is converted oxidized into NAD positive, the enzyme which is used is dehydrogenase. Secondly, whenever there is a shift of functional group from one position to another, for example, here we have carbon chain, and if phosphate is present on the carbon number 3 and it shifts to carbon number 2, there is a shift in the position of phosphate from carbon number 3 to carbon number 2. Whenever this shift occurs, the enzyme mutase is used. And the third thing is, except the rate regulatory steps of glycolysis, which are 3, all other steps are reversible, which means they can proceed both in forward and reverse direction. Now, continue the process of glycolysis. We form glucose 6-phosphate from glucose, and then fructose 6-phosphate is formed from glucose 6-phosphate, and then fructose 1,6-bisphosphate is formed from fructose 6-phosphate. Now fructose 1,6-bisphosphate is cleaved into two trioses, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And the enzyme of this reaction is aldolase. Aldolase enzyme is used here. These two are trioses. Trioses mean which contain 3 carbon unit. Fructose is a 6 carbon carbohydrate, whereas each of these are 3 carbon carbohydrate. This 6 carbon fructose is cleaved into 3 carbon glyceraldehyde and 3 carbon dihydroxyacetone phosphate. Now to further proceed the reaction, this dihydroxyacetone phosphate is converted into its isomer. Glyceraldehyde 3-phosphate is isomer of dihydroxyacetone phosphate, and it is reversibly converted into its isomer. And as I told you earlier that whenever isomerization occurs, enzyme isomerase is used. Enzyme isomerase is used here, and as it is triose, so triose phosphate isomerase enzyme is used here. Initially there is single glyceraldehyde 3-phosphate, but when dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, now 2 glyceraldehyde 3-phosphate are formed. And now onward the reaction proceeds in the form of 2. Now the next reaction is the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This is the first oxidation reduction reaction of glycolysis. Initially only phosphate is present only in carbon number 3 of glyceraldehyde. Now on carbon number 1 another phosphate is present, so 1,3-bisphosphoglycerate. And as this is the oxidation reduction reaction, NAD positive is reduced into NADH. And as I told you in the start of the video that whenever oxidation or reduction of NAD positive occur, the enzyme dehydrogenase is used. And as this is the glyceraldehyde 3-phosphate, so glyceraldehyde G3-pre, glyceraldehyde 3-phosphate dehydrogenase enzyme is used in this reaction. Now there are a few things that you need to learn about this reaction is that this phosphate, which is attached at carbon number 1, is attached through a high energy bond. And this high energy bond is used to synthesize ATP in the next reaction of glycolysis. So you came to know that in the next reaction ATP is formed. Secondly, NAD positive is present in limited amount in the cell. And if all the NAD positive is converted into NADH and can't replenish, then NAD positive becomes shorter in the cell and glycolysis can't proceed further. So for the continual supply of NAD positive, NADH is converted into NAD positive, first when pyruvate is converted into lactate, and second in the ETC, electron transport chain. But the problem here is NADH can't cross the inner mitochondrial membrane of mitochondria to enter the mitochondrial matrix and to participate in electron transport chain. So NADH enter the mitochondrial matrix crossing inner mitochondrial membrane by using marate, aspartate shuttle or glycerol 3-phosphate shuttle. We will discuss it in detail, but ये आपके book में mention था, so मैंने आपको बता दिया ताकि आपको confusion ना हो कोई. So the next thing about this reaction is in RBCs, this 1,3-bisphosphoglycerate, which is the product of this reaction, is converted into 2,3-bisphosphoglycerate by using the enzyme mutase. Why enzyme mutase is used here? As I told you in the start of the video that whenever there is a shift in the position of functional group, enzyme mutase is used. And here you can see that first initially phosphate is present at carbon number 1 and now phosphate is present at carbon number 2. So there is a shift of functional group, so the enzyme mutase is used here. And as this is phosphoglycerate, so enzyme phosphoglycerate mutase is used. This 2,3-bisphosphoglycerate in RBCs for oxygen delivery as you studied in first year. Now this 2,3-bisphosphoglycerate is converted into 3-phosphoglycerate by using the enzyme phosphatase, because you can see that one of the phosphate is removed. And as I told you earlier that whenever dephosphorylation occurs, enzyme phosphatase is used. So here enzyme phosphatase is used and it is converted into 3-phosphoglycerate. So this is the reaction occur in RBCs. This is not the part of the process of glycolysis, just it is mentioned in your book and it is linked here, so I told you. Now we continue the process of glycolysis. Now this 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and as it is not the rate regulatory step, so it proceeds both in forward and reverse direction. And the enzyme used here is kinase. Why enzyme kinase is used here? Because the phosphate in this 1,3-bisphosphate is transferred to ADP, and so ADP is converted into ATP. The enzyme kinase is used here because phosphorylation of ADP occur and ATP is converted into ATP, and the phosphate is provided by the substrate. Full name is phosphoglycerate kinase, and it is substrate level phosphorylation because the ATP formed here is not through oxidative phosphorylation, but for the formation of ATP, phosphate is provided by the substrate. So this is substrate level phosphorylation. Now this 3-phosphoglycerate is converted into 2-phosphoglycerate, and the enzyme used here is mutase. Why enzyme mutase is used here? Because phosphate initially present carbon number 3 is now shift to the carbon number 2, so the enzyme mutase is used here, and of course the full name is phosphoglycerate mutase. The next step is conversion of 2-phosphoglycerate into phosphoenol pyruvate. Dehydration of water occur, which mean water is released, and the enzyme used here is enolase. ये आपको याद रखना पड़ेगा, ठीक है. तो 2-phosphoglycerate phosphoenol pyruvate में कन्वर्ट हो गया बाय यूजिंग द एंजाइम एनोलेज. हाँ, ये जो फॉस्फेट इनोल के साथ अटैच है ये एक हाई एनर्जी फॉस्फेट है, और ये हाई एनर्जी फॉस्फेट नेक्स्ट स्टेप में ATP बनाने के काम आएगा. सो वी नो बिफोर हैंड दैट नेक्स्ट स्टेप ATP इज फॉर्म्ड.